Hello, and welcome to this week's edition of Your Class. Today, there's two things on my agenda. Number one, I wanted to talk briefly about what you can expect when it comes to material from week to week. And number two, I'm going to actually jump into some of that material. So, number one, I wanted to reiterate, I know I have written it down a lot in each of the modules. However, I need to reiterate that it is important that you read each of the chapters that is assigned in each class. I will be going through and taking out pieces of the material and teaching you about it individually. Especially if I think that there's a certain part that is very confusing, I'll try to kind of prioritize that part. I'll also use supplementary videos. This is designed to kind of activate other parts of your brain when trying to digest the material. However, it would be a mistake to assume that if you only went through my videos, the PowerPoints that I'm making, the supplementary videos, and did not read the chapters, yeah, that would be a mistake. It is important for you to read all of the material. When you do see my PowerPoints that are on there, some of them I will go in and make a video for and explain. Other ones have been designed so that they are like reviews. They are set up to be pages of material that is reviewing material from your text, hopefully in a way that might be a little bit easier to digest, but it is all from your textbook that I've gone through and tried to make look a little different, feel a little different, maybe it'll go down a little bit easier. So yes, of course, use it all, watch all the videos, go through all the PowerPoints, all of that's good stuff. Just don't forget to also read the material in your book. It's all supposed to go together. It's not an exclusionary thing. Okay. Now that I've said that, I want to jump into the material that I've decided to go over this week, which is, at first hearing of it, probably sounds a little intimidating, but I'm going to jump into genetics. So I actually really love this material. I love genetics. I know it seems strange. One reason why I think I love it so much is it's because it's the first class I ever got an A+ in at UCSD, and I'm still proud of that to this day. However, there's also just something about genetics that makes sense. Now, I'm not somebody who particularly likes math, but I do like symbols and I do like puzzles. And if you understand the symbols, then genetics can kind of start to feel like a puzzle that you can put together and make sense of, which is even better because I love to try to make sense of things. So if you're trying to figure out genetically what's going on with you or how something works, it's actually sometimes pretty straightforward. And I'm going to go through some of that with you today. Another reason why I love genetics so much is, as some of you may remember, I, as a side business, breed snakes. And to get into breeding snakes, genetics is super helpful to figure out exactly what you are doing, your game plan of who you're going to breed with who. For example, if I were to take just two normal snakes, so I breed Kenyan sand boas, and the ones that you would find in the wild, the way that they look, they're called normals, normal presenting, right? If I were to breed them together and I didn't know what was going on genetically, I might just continue to get more and more normals, which sure might be cool. I mean, they're good little snakes. However, they're going to run me $75 a snake, which at about 20 snakes in a clutch or a litter, as they call them, would be decent, sure. However, if I know genetics and I know, well, if this snake is heterozygous for this and this one's homozygous for that, and I put these ones together, then I can actually get a litter of snakes that's closer to $750 per snake times 20. So you can see how understanding the basic form of genetics that I'm about to go over with you can be fun, can be financially helpful, can help answer questions about who we are and what's kind of going on within our system and our genes, and can kind of make a lot of sense with stuff. So let's jump in, okay? Let me share my screen. This is actually the second time I've made this recording, because the first time I clicked on the wrong button. So, second time's a charm, right? Okay. Let's hope this one goes better. All right, so welcome to Genetic Foundations. Now, most of this can all be found in your textbook. There are a few pieces on here called the Punnett square that is a little bit new, but the concept is all described in the text. I just kind of describe it a little bit differently because it might make a little bit more sense my way. Again, this is at least the way my brain works. Maybe yours will too. So we're going to jump in. Recognize that a lot of what we're going to go over in the very beginning is kind of review, so I'm not going to touch everything in this slideshow because some of it you probably should already know, especially if you were one of those people, and there's about like 5 of you who have taken my general psychology class, and I know I have taught you this before. So hopefully this will be a review. If not, I guess I'll never know, right? You'll learn it this time. So for this reason, I'm only going to minimally touch on some things, most of those things being terminology, but we will kind of jump into it a little bit too. Okay. So when it comes to terminology, first thing we have chromosomes. What's a chromosome? Now, do you remember? I hope so. So I know I have the answer right smack on here, but do you remember how many pairs there are of chromosomes? 23. Good job. So we have 23. Now the very last pair, that 23rd pair, do you remember what that 23rd pair was? Huh? Starting to jog your memory a little bit? Okay, well, what about this question? Do you remember what is indicative of a pair of XX or XY? What these would mean? Right? Because it is the same thing. The 23rd pair and these chromosomes right here are the same, and they are indicators of chromosomally, I may be making up that word, what a person is, what sex that person is, whether they are chromosomally female or male. Do you remember which is which? Which one's XX, male or female? XY. Right? Hopefully jogging your brain in here. If you were to have a question on here, I'm hoping that you would be able to answer. If you need a little bit of reminding, XX is the sign language for X. XX is indicative of a female, and XY, sign language for Y, is indicative of a male. Okay, so DNA we should know, genes you should know, gametes and zygote. There's some videos also posted for you that will review those things as well. Okay, multiple offspring. So here's another question for you. What is the difference between a fraternal twin and, typo, and identical twin? What's the difference? Is there a difference? Yeah, there is. What are the similarities? Well, one of the similarities is twins are born generally at the same time. They are incubated or gestated at the same time, right? They're going to be in there. However, when it comes to how this came about, it is going to be different. So what about this question for you? When it comes to fraternal twins, is there a difference between two fraternal twins and two siblings genetically? Any difference there? Are fraternal twins any more like each other than siblings? And some of you may realize this already, that is a trick question. No, they're not. They're not any more similar. They are the exact same genetic difference. The only difference between a fraternal twin and a sibling, like a brother or a sister, is that fraternal twins were just incubated and born at the same time, versus brother and sisters probably has like 2 years or something like that between you at least. That's probably an assumption right there. That's me and my sister, I should say. But genetically, fraternal twins are just as different as siblings. They are. It's might as well have your sister or your brother or whoever born at the same time as you, and you would be the same genetic difference as a fraternal twin. Now, what about an identical twin? How is that different? Right? If I were to say, okay, when it comes to fraternal twins and identical twins, how many eggs are involved? Yeah. Of the fertilization process, sperm. Yeah. So for a fraternal twin or any sibling, there's an egg for each person that is going to be born and a sperm for each person that is going to be born, right? Exact same thing as siblings. Now, identical twins, that's not the case, right? There's different ways that identical twins can be born. The most common one is there's one egg, one sperm, fertilization, and in the process of the development of this zygote, it actually breaks apart into two. And now you have two separate yet identical fertilization processes happening, right? Two different development, two different people, however, genetically identical, right? Alleles. Alleles are going to come up pretty soon. The biggest thing to know about an allele is that you, for every gene, get an allele from each parent. I'll talk about that. Genotype versus phenotype. What are the differences there? So a genotype is going to be genetically what's going on, right? What genes did you get? You may not see what genes you have, right? Versus a phenotype is what does it look like? Did it present? Do you see it? And if you see it, that's that phenotype, okay? So for example, I'm just going to talk about my snakes. One of the things that I can go for when I am deciding what I want to go for is albinism, having a snake that's an albino, right? So that's going to be a recessive gene, which we're going to talk about, but it would look like an albino. That would be the phenotype, versus a genotype may be different. They may carry the albinism gene, however, they don't look like it, right? So that's going to be a genotype versus a phenotype, what they look like versus what their genes are. Homozygous and heterozygous. So that first part, homo, means same. That first part for the second one, hetero, means different, okay? So homozygous means that the alleles, right, are the same. Okay, so they got the same thing. Versus heterozygous means that they got different ones, essentially. It's not identical. Okay? Sometimes in the industry, we'll call it het for short. Instead of heterozygous, we'll just say het. And you'll see where that comes into play later. Okay. So the biggest kind of inheritance that we're going to talk about today is the dominant recessive inheritance. Okay, meaning that when a gene is expressed, there are dominant pieces and recessive pieces. Usually the dominant pieces are characterized by capital letters and recessives are characterized by lowercase. Now, if there is ever a dominant piece to a gene there, like let's say you got a dominant part of the gene from your dad, then the hidden gene, the recessive gene, isn't going to be shown. A dominant gene always cancels out a recessive gene. Always is a bit of a stretch, but let's just say for these dominant recessive examples, this is true because it most of the time is. So the example that we're going to use instead of snakes is PKU. So this is something that can impact people. So having PKU looks like this. It will affect the way the body breaks down proteins contained in many foods. Babies who are born with PKU lack an enzyme, and this can lead to, can't say that word, for this enzyme to build up to a toxic level that can damage the central nervous system, causing permanent intellectual disability. So if for some reason a baby is born with PKU and they eat a certain kind of food, it will build up a toxin in their brain and make them intellectually stunted. This is no good. This can cause major, major lasting effects. Now, if we are able to test for it, which we can, and we catch it early, and we just tell the parents of that child, well, infant, not to eat those certain kind of foods, their brains never form that intellectual disability. So it is a good thing for us to be able to track this because we can literally save lives. So let's use this PKU example. Now it is an example of a dominant recessive inheritance. Okay. So I know there's a lot to unpack here. Here are my letters, here are my symbols that I was talking about earlier. So let's pretend that these two up here, everything that's up here is going to be the father, and everything on the side here is going to be the mother. Everything within the box is going to be their potential offspring. So statistics, kind of, these are the chances of this couple producing this kind of offspring. So you can see, because you're always going to grab one allele from your dad and one allele from your mom, there's always going to be kind of these 4 different options that can happen. Okay? I know that right now they look identical, and I'll explain it. So let's pretend that your dad for PKU has completely dominant alleles. Now remember, dominant alleles can be seen in the capital letters. So I've just used B because B for Brianna, B for be yourself, B for best. I don't know. I decided to go with it. So let's pretend that dad has a dominant PKU gene, meaning that both of his alleles are dominant. And let's pretend that mom has the same. So both of her alleles are dominant. Okay? Remember those 23 chromosomes, right? Everybody gets one from mom, one from dad. I know I'm saying that a lot. So what's going to happen is, here are the chances. This B will go here. This B will go there. Cool. This B here. This B there. This B here. This B there. This B here. This B there. So again, now you can see all the possibilities between this couple and their offspring. All of their offspring look the same as they do. They are all dominant for not having that PKU. Remember, if they even have one capital B, one dominant letter, it wipes out the PKU. They're not going to have it. Not only that, but they will not be able to pass it on to their offspring. Okay, so no PKU, only more dominant genes for no PKU. So that one's pretty straightforward. And remember, when they look the same, like this capital B and capital B, that's going to be an example of a homozygous dominant trait. All right, what if it looks a little different? So this time, let's pretend that dad is actually a carrier. Okay, so genetically, he has one of these lowercase letters, right? But phenotypically, he does not have PKU. He doesn't look like it. He doesn't have it. He doesn't have the issue. He just happened to have got one of the recessive genes from one of his parents. But again, this capital letter, this dominant part of his alleles that he got, wipes out the lowercase so you don't see it. However, he has the possibility of giving it to his offspring. So again, pretending that dad is these 2 letters, capital B, lowercase b, and mom hasn't changed, mom is still both dominant, we would do the same thing, right? So this B here, this B there, this B here, this B there, this B here, this B there, and this B here, and this B there. Now, you can see the possibility of their offspring. About 50% have both dominants, right? So just like last time, they will be unable to pass along the PKU. The carrier part from the father got knocked out. It is no longer in existence. It is gone in his offspring. However, with the other 50%, they have, yes, a dominant gene, but they also got that carrier gene here. So they will not have PKU because the dominant gene knocks out the lowercase, the recessive, right? However, when they have offspring of their own, there will be the possibility of them passing along this lowercase or recessive part of the allele. Okay, so there's no PKU. None of these offspring would have it because they all have a capital B somewhere, which again, the dominant would be stronger than the recessive. It knocks it out phenotypically. But 50% of the children would be heterozygous or het for PKU, meaning that they're carriers. Okay. I hope this is starting to make sense. With more, it usually gets a little bit easier. Okay. This time, let's pretend that both dad up here and mom down here are both carriers. Again, not phenotypic. They do not have PKU. They're only carriers, right? Because they do have this dominant gene. And the dominant gene knocks out the ability for this lowercase one to kind of take over, right? But let's see what happens statistically with their offspring. So this be here, this be here, this be there, 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 and this be there. Uh-oh, well, this one looks a little different. Number one, I put it in red, which is usually scary, so you're kind of getting a hint that something's going on there. But also, we have both lowercase b's, meaning both of these are recessive. If both are recessive, the recessive phenotype is going to show. So there is a 25% chance, or 1/4 of a chance, that if two parents who are carriers have offspring, that the offspring will have that recessive gene. And if they inherit both of the recessive genes, they will have PKU, or whatever thing we're talking about with this, because this is, again, used in a lot of different examples. Okay, 50% have a uppercase and a lowercase. Remember, that means carrier, right? Or heterozygous. And another 25%, or 1/4, have both big B's, meaning that the carrier gene got knocked out completely, and they won't be able to even pass it on to their offspring when they have kids, right? So both capital B's and lowercase b's are both homozygous because they are the same. One is homozygous dominant because they're capital. One is homozygous recessive because they're recessive. And these two would both be heterozygous because they are different. So again, 25% of a chance of the children having PKU, 50% heterozygous for PKU, and 25% chance fully dominant against PKU. Now, this is the exact same Punnett square that I use for my snakes and albinism. Normals are these capital letters. Albinos are these lowercase letters. So if I'm taking two normal looking snakes, but I know that they're heterozygous, and I breed them together with each other, I know that I'll have 1/4 of a chance of producing an albino. So let's say I had 4 snakes, 4 snake babies. Oh, one of them would be albino, 2 of them would be heterozygous, and 1 of them would be straight up dominant normal. Okay? So it all kind of goes about the same for certain genes. Okay, let's look at the last example. Recessive homozygous. So let's pretend both dad and mom have PKU, right? So they do not have a dominant gene. They both have these recessive genes for both of their alleles. Now, when they pass them to their children, you can see B goes there, 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 B goes there. 100% of their children will also have PKU. So if you, for example, had or have PKU and your significant other does too, and you have children, they will as well, 100% of the time. If you were to take two albino snakes, remember they're also both recessive. Dad snake, mom snake, 100% of the babies from the albino snakes would be albino. Kind of a good deal if you have two albinos, right? Not a good deal if it's PKU. Okay. So here's the picture that is in your textbook describing this. It's figure 2.3 of dominant recessive mode of inheritance illustrated by PKU. So we have the father here and the mother, both of which are described as carriers. And then we have the four possibilities of children. Now remember, statistics. So we're statistically assuming what would happen. A fourth of their children would have that dominant gene, so they would not get the PKU and they would not become carriers. 50% here would become carriers, but they wouldn't show it. And 25% or 1/4 of them is going to have both recessive, meaning that they're going to have the PKU. In this picture, instead of an uppercase and a lowercase, they're using N and P. Probably for no PKU and PKU, I'm assuming. Same difference. Okay. So that is a dominant recessive gene. Not all genes are like that. For example, we have this one that's called an incomplete dominance pattern. And an example of this is sickle cell. Now, sickle cell is a really terrible genetic disorder that is still, unfortunately, very much an issue. So it acts very much the same way as PKU, but it has some exceptions. So if an individual who is heterozygous, so carrier, right, for sickle cell experiences oxygen deprivation, the single recessive allele can assert itself and a temporary mild form of the illness can occur. So what does that mean? So what happens with sickle cell is the cells are shaped like sickles, your blood cells are shaped like sickles as opposed to like a round cell, like cells usually look. And what can happen is when they're shaped like that, they can clog each other really easily. They also have a harder time carrying oxygen to different parts of the body. So you can imagine if somebody has cells that literally makes it harder for them to carry oxygen around in their blood, it would be bad if they experienced oxygen deprivation. So let's say you didn't actually have sickle cell. You don't have the two recessive letters on there. However, you are heterozygous. You're a carrier. What can happen is, let's say you went really high up in a plane, or you went hiking and you went up to a really high elevation or something, and you experienced oxygen deprivation. Now, if you're heterozygous for sickle cell, what can sometimes happen is that little recessive gene that usually would get knocked out by the dominant one can stick up for itself, and it basically takes over as if you actually had two recessives, and you would experience a mild form of the symptoms of sickle cell, right? So you wouldn't be having a good time, and you would experience it temporarily as long as you were in that oxygen deprivation stage. Now, once you were able to like come back down the mountain or whatnot and you're able to recover, it would go back to being a hidden thing. You wouldn't know about it. But that's why this is called an incomplete dominance pattern. Otherwise, it would work exactly the same way as PKU. If you had two recessives, you would have sickle cell. If you are heterozygous and you have one dominant, one recessive, then you're a carrier, and that's it. Unless with this exception, something happens in your environment. Or you could be dominant, right? And where you don't even have the recessive gene involved, okay? Or allele involved. Here's another one for you. So these are called X-linked inheritance, okay? So if you remember from earlier, the sex chromosomes that we have women as being XX, and we have males as being XY. So when at first you see X-linked pattern problems, you might want to assume that women would have a bigger issue with it because we have two X's, right? Where the males only have one. But if you think about it a little bit further, the males get basically a 50-50 shot of inheriting a recessive X-linked issue, right? They get one X from their mom or from their dad, and that's it. So essentially, if mom or dad is a carrier for an X-linked issue, the offspring's going to have a 50% chance of having that same issue. Versus females get an X from mom and an X from dad. And if one of them is dominant against an X-linked allele, it'll knock out the other one and you won't see it, right? So you can have two dominants. You can be heterozygous, and remember, heterozygous, you wouldn't have any issue. You would just be a carrier because that dominant one would knock out the effects of the smaller one. And if you were a female and you had an X-linked genetic issue, that means you got really unlucky, and from both mom and dad, you got both little recessive ones. So they weren't able to knock themselves out. But you can again see that females have less of an opportunity to inherit these issues, right? There's 3 different ways that they can express it genetically, 2 different ways that they can express it phenotypically, versus males genetically, they only have 50-50. So an example of this would be hemophilia, another issue with blood. But I believe another example that you may be more familiar with is colorblindness. This is why more men are colorblind than women. They have more of a chance of inheriting it because it's on that X-linked issue. Okay. Here is a figure from your textbook, figure 2.4, X-linked inheritance. Right, and you can kind of see the same thing that I just explained, but kind of explained in a different way. I'm not going to go over this. I feel like you can kind of get a little bit more information from your textbook on this one. The last thing that I kind of want to talk about in this presentation is that's pretty simple and straightforward, what I just explained. Not all genes are that simple. Okay, so the word poly, if you ever see that in a word, that means many, right? So polygenetic, many genes inheritance, meaning that there's many different factors that can go into certain aspects of a human. So some of those things are things like height, weight, intelligence, personality. It's not just one gene that goes into creating what this is going to look like for you. There's going to be many. So again, many genes affect the characteristic in question, not just the one like we just looked at. Those ones are much more complicated. Those ones are not ones that I am going to explain to you right now. And that's it. All right. Well, I hope that you had some fun learning a little bit about something that I'm pretty passionate about, that you feel a a little bit smarter when it comes to genetics, that maybe some of that information that you had kind of learned before sunk in a little bit differently this time, and that you can maybe even use this in your everyday life whenever you want to talk about being a geneticist or something. Other than that, I hope you had some fun. I certainly did, and I will see you in cyberspace. Okay. If you do have questions, come see me on my office hour, Mondays. Good times. I would love to talk about it. I obviously love to talk. Yeah, I'd love to meet all of you. Okay. I'll see you around.